Now we are going to study Hybridoma technology. What is Hybridoma technology? It is a method used for the production of monoclonal antibodies. For the production of monoclonal antibodies, Hybridoma technology used. Firstly, immune cells are used for the production of polyclonal antibodies. This technique is discovered by G. Kohler and C. Milstein in the year 1975. In this technique, hybrid cells are produced. From the hybrid cells, monoclonal antibodies are produced. So, hybrid cells is produced by fusion of B lymphocytes and myeloma cells. These myeloma cells are responsible for. These cells have the property of rapid uncontrollable divisions. The properties of these two cells occurs in this hybrid cell. Firstly, we need to learn the definition. So we learn what is hybridoma technology. It is a method used for the production of monoclonal antibodies. And then with production, purification, and application. The production of monoclonal antibodies, the production involved in the hybridoma technology is immunization, cell fusion, selection of hybridomas, screening the product, cloning and propagation, characterization and storage. Immunization is nothing but B lymphocytes production, and myeloma and B lymphocytes are fused, and then different types of fused cells are found. In that, hybridoma cells are selected, and then from the hybridoma cells, monoclonal antibodies are collected. These are again cloned and propagated. Then for the characterization, storage is done. First is immunization. So this is the rat. In the immunization process, we make rat to develop B lymphocytes. So these are the lymphocytes obtained from the spleen of rat, and then we use myeloma cells, and these both are fused with the help of HGPRT. These 5 different types of cells are formed: unfused, fused myeloma cells, unfused myeloma cells, hybridoma cells. These are found, but hybridoma cells are necessary. We select these hybridoma cells and we introduce them to HAT media. All are introduced in this HAT media. Only hybridoma cells can survive. After this, and from hybridoma cells, monoclonal antibodies are produced, and these are harvested and storage. After harvesting, purification is done using centrifugation and filtering, then storage is done. So the immunization technique involves the following steps. Firstly, we select an animal, that is mouth, which should be healthy, disease free, and we should check its weight. The animal should be healthy, disease free, and we should check its weight. Then antigen is injected to the mouse through subcutaneous route initially, and then multiple doses given. Now, we should select an antigen and inject it to the mouse through subcutaneous route initially for once a day, and then multiple doses given. 3 days before sacrificing the animal, dose is given intravenously, and frequency is dose also increased. It is done so that number of antibodies are produced. We should do this procedure until we should do or inject the antigens until required antibodies are produced in the rat. Then we should extract the antibodies from the rat sacrificing it, and we should perform ELISA test to know the activity of the antibodies. Then we sacrifice the animal and spleen is separated. From the spleen, lymphocytes are extracted by density centrifugation of spleen cells. By performing density centrifugation of spleen cells, lymphocytes are extracted. Then these lymphocytes are washed and fused with myeloma cells, and during fusion HGPRT is also added. HGPRT is nothing but hypoxanthine guanine phosphoribosyl transferase enzyme is added to help the growth of hybridoma cell. Hypoxanthine guanine phosphoribosyl transferase enzyme is added during the fusion. And after that, PEG is also added to remove the toxicity. After some time, it is washed to remove the PEG. Then after beta cells has fused with myeloma cells, hybrid cells are produced. Actually, 3 different cells are produced: unfused plasma, fused plasma, hybridoma cells, fused myeloma, unfused myeloma. These are the 5 different type of cells, but we need hybrid cell. In order to have a cell, DNA should be present. So for the synthesis of DNA, there are 2 processes, salvage and de novo process. In the salvage process, new nucleotide is produced using an old nucleotide. That is, old nucleotide is a precursor. In this process, HGPRT helps in this process. While in de novo, amino acids and sugars are required for the production of nucleotide. But here in this technique, salvage process is only used. That's why we add HGPRT while fusion. All the 5 or only hybrid cells are taken and transferred into HAT media. HAT means hypoxanthine amino protein thymidine media. Hypoxanthine amino protein thymidine media. In this media only allows the growth of hybridoma cells. Only hybrid cells will survive, and these cells develop monoclonal antibodies. These are harvested. So, in this harvesting, antibodies are separated from the contents of the media. Then screening is done. Screening is done to check the capability or activity of monoclonal antibodies. For screening, ELISA or RIA tests are done. And then cloning or propagation is done. The MABs having capability are separated and cloned for the further more production of monoclonal antibodies. Cloning is done by 2 steps: limited dilution method or soft agar method. In limited dilution method, microcultured wells are utilized. Hybrid cells are suspended in microculture wells. In the soft agar media, semi-solid media is used. Then monoclonal antibodies undergo propagation, that is multiplication. After that, characterization of MABs are done. Characterization is done based in terms of biological and physical activity. So that is, biophysical characterization is done to check its working. If it passes, then it undergoes storage. This is the process of. But before storage, purification is done. Purification is done to separate antibodies from its cell content, clotting factors, etc. Harvested after purification, they are centrifuged and filtered for the purification. Filtration is done using 0.45 micron filters. Now the applications are diagnostic purpose, that is biochemical assay and diagnostic image. Therapeutic purpose, protein purification, miscellaneous uses. Firstly, diagnostic purpose. In the biochemical assay, to perform the tests like ELISA and RIA, MABs are used as reagents. And in diagnostics imaging, MABs are used as radio-labeled substances. And in therapeutic purpose, for the direct action, MABs are used, and also for the targeted action. In the direct action, MABs directly acts against disease-causing organisms, example cancer, AIDS. And in the targeted action, MABs are used as target antigens. We use MABs for drug delivery, used in cancer, radioimmunotherapy, and they are used for the protein purification. Miscellaneous uses are MABs also have enzymatic properties, so there may be catalytic MABs, also autoimmune MABs are also there. They are used for autoimmune diseases and also help in identifying fingerprint. These are the applications. Diagnostic, therapeutic, protein purification, miscellaneous. In the diagnostic, biochemical assay and diagnostic image. In biochemical assay, tests are performed in which MABs are used as reagents. In diagnostic image, MABs are used as radio-labeled substances. In therapeutic purpose, there is direct action and indirect action. In direct action, that directly acts against the disease-causing organisms, example cancer, AIDS, etc. And in the targeted action, MABs acts as targeting agents. They're used for drug delivery, radioimmunotherapy, also used in cancer. They are used for protein purification. Miscellaneous uses are they have enzymatic properties, so the substitute enzymes have these are also auto antibodies. We can identify auto antibodies and help identify fingerprinting. These are the applications of hybridoma technology, or monoclonal antibodies. And now we will also again revise it faster. Hybridoma technology is nothing but for the production of MABs. This method is used. Immune cells. If we use immune cells for the production, they are called polyclonal antibodies. G. Kohler and C. Milstein discovered this process in 1975. From hybrid cells, MABs are produced. Hybrid cells are obtained by fusion of B lymphocytes and myeloma cells, two characters in one cell. Production involves immunization, cell fusion, selection of hybridomas, screening the product, cloning and propagation, and then characterization and storage. First, in the immunization, mouse which is healthy, disease-free, healthy weight is taken. Antigen is inducted first through subcutaneous route initially, and then for multiple doses. Then 3 days before sacrificing, dose is given intravenously and multiple times, so the number of antibodies produced. Then it is screened or assay is performed to check its antibody. Then animal is sacrificed and spleen is separated from the animal. Lymphocytes are extracted from the spleen by density centrifugation. Lymphocytes are washed and fused with myeloma cells. HGPRT is added. HGPRT is nothing but hypoxanthine guanine phosphoribosyl transferase. This is used for the salvage process. Then PEG is added to remove toxicity, and then it is also washed. 5 different types of hybrid cells are obtained. And for the production of hybrid cells, salvage and de novo processes are involved. In salvage, old nucleotide is used, while in de novo sugars and amino acids are used. But we use salvage in this process. HGPRT helps in this process. Then hybrid cells are transferred to HAT media, hypoxanthine amino protein transferase media. Only hybrid cells are survived. Monoclonal antibodies produced, harvested, separated from cells and other media contents. They are screened by assay and RIA. These methods, ELISA and RIA assay methods. Then cloning and propagation is done. Cloning is done based on limiting dilution or soft agar method. In limiting dilution, microbe culture wells are used. In soft agar, semisolar solid media is used after propagation. Biophysical characterization done, and before storage, purification is done. Purification is done by separation of antibodies from cell contents, centrifugation and filtered using 0.45 microns. And then applications, diagnostic, therapeutic, protein, miscellaneous. Thank you.